Hello everyone, welcome back. Today we'll talk about neoplasia, and as you see, neoplasia, the term has two parts. The first part is neo and the second part is plasia. The first part neo means new and the second part plasia means growth. So neoplasia is actually new growth of tissue, and this growth has certain characteristics, and all of them, the most important is the growth is excessive due to unregulation, and the growth is autonomous. So there will be excessive autonomous growth, and which is not reversible, and which is also monoclonal. Monoclonality is something like that. Suppose this is the tissue, and in the tissue there is one cell, and this one cell gets abnormal, and this one cell starts to divide abnormally, excessively, and it gives rise to 2 cell, 4 cell, then 8 cell, and it goes on. So it's actually monoclonal. So every cell is actually started off from one cell. Okay, so this is monoclonality. Okay, so the basic types of tumor are benign and malignant tumor, and benign tumors are good and malignant tumors are bad and ugly. I will explain later why I am telling so. Okay, so the nomenclature of tumors depends on the lineage where actually the tumor came from. If the tumor came from epithelial tissue, the benign tumor will be called oma. So it's a suffix which will be added on the last part of the tumor name. So adenoma is a benign tumor of the glands. Papilloma is a papillary-like structure which is also a benign tumor, and the malignant counterparts will be called carcinoma. So if the adenoma is the benign tumor, its malignant counterpart will be adenocarcinoma. If papilloma is the benign tumor, its malignant counterpart will be papillary carcinoma. If the tumor is from mesenchymal origin, also the benign tumors will be called oma, but the malignant counterparts changes its name and they are called sarcomas. So suppose a benign tumor is fibroma, benign mesenchymal tumor, and its malignant counterpart is fibrosarcoma. And benign tumor is lipoma, malignant counterpart is liposarcoma. Okay, there is some special things to know about this nomenclature. There are some oma which are not benign, and those are lymphoma, seminoma, and some forms of meningioma, glioma, and even hepatoma, though melanoma. Those all omas are omas, but they are not benign, they are actually malignant tumors. So we must know what are the common tumors, how common it is, and it's the second leading cause of death in adults and children worldwide. And the first leading cause of death is cardiovascular disease, and second leading cause is cancer. And among the cancers, according to incidence, the most common cancers in male is prostate cancer, in female is breast cancer. But according to mortality, the fatal cancer is lung cancer. Okay, so the other cancer is colorectal cancer, which is a third in both lists. Okay, so we have a big question here: why cancer develops? And for this big question, there is a short answer: simply mutation of DNA. So this mutation is not a normal mutation. If a mutation occur, not always it does develop into a malignancy. So this mutation should involve some special genes, and those are genes which actually regulate cell growth. So those all genes are regulator of cell growth. So if any of those are mutated, it can lead to an excessive growth. So those are proton cogenes, tumor suppressor genes, and regulator of apoptosis. I'll talk about those in later slides. So let's first look about why mutation occurs, or who actually causes mutation. So mutation can be caused by ingestion or inhalation of some chemical agents, or infection by a virus, or even radiation exposure. Okay. So chemicals are the most common ones, and among the chemicals, cigarette smoking is the #1 cause of chemical induced mutation and leading to cancer. And cigarette smoke actually contains some chemical substances called polycyclic hydrocarbons. Okay, the arsenic is also a chemical carcinogen which can cause skin cancer, lung and lung cancer and vascular cancer like angiosarcoma. Nitrosamines are some chemical substances found in fishes, such as salted fishes eaten by Japanese sushi. Okay, so those can lead to stomach cancer. Alkylating agents, those are actually chemotherapeutic agents used to treat different type of malignancies like leukemia, lymphoma. So the fun thing is those are used to treatment of lymphoma, used in the treatment of lymphoma, but they can also give rise to leukemia and lymphoma. So that's a very bizarre thing. Alcohol is also a chemical carcinogen which can cause cancer in the esophagus, and pancreas, or liver. And also there are other two are vinyl chloride associated with hepatic angiosarcoma and naphthalamine associated with bladder tumor or bladder cancer. Viruses, the most important viruses to know are hepatitis viruses, which causes hepatic carcinoma or hepatocellular carcinoma. HPV or human papillomavirus, especially strains 16, 18, 31, 33, all of them are associated with cervical carcinoma. Epstein-Barr virus causes nasopharyngeal carcinoma, Burkitt's lymphoma, and CNS lymphoma in case of AIDS patient. Human herpes virus 8 can cause Kaposi sarcoma, especially in case of an AIDS patient. Human T-cell leukemia virus actually causes leukemia. It's so simple. And radiation. So there are two types of radiation, ionizing and non-ionizing. The most common form of ionizing radiation is X-ray or radiotherapy, which can actually cause leukemia and also thyroid cancer. And non-ionizing radiation, the most common example is ultraviolet B spectrum from sunlight, which will actually fall into skin and can cause skin cancer. So as we have previously mentioned, that the cancer develops due to mutations, and those mutations can involve one of three different classes of growth regulators. So first class of growth regulators are proto-oncogenes and oncogenes. So if you look at the name oncogenes, okay, or proto-oncogenes, so you have the onco portion in the incorporated in the names. So actually those are the genes which actually favors growth. So actually every one of us, every human being has proto-oncogenes. Those are essential for cell growth, but those are regulated growth. So this growth is controlled. Okay, but when these proto-oncogenes are excessively activated, this transform into oncogenes, and those oncogenes will lead to excessive growth more than normal, leading to formation of a malignancy. Okay, so if we consider how growth occurs, what are the mechanism that a cell needs to be grown, is like that. Let's draw a picture here. Suppose this is a cell. This cell has a receptor here for growth factor, and here is a growth factor. So the first thing is, for growth of a cell, a growth factor is needed to bind to the growth factor receptor, and after the binding the receptor, there is activation of the receptor and the signal transduction pathway, and after the activation of signal transduction pathway, it will lead to activation of nuclear submaterials, DNA activation, and leading to ultimate proliferation of the cells. Okay, so we can actually mutate any part of this system. We can actually have increased amount of growth factor or abnormal growth factor, an abnormal growth factor receptor, or abnormal signal transition pathway, or abnormal nuclear factor. Okay, so here we have. So the growth factor can be excessive, or growth factor can abnormally activate some growth factor receptors, such as platelet-derived growth factor beta or B is associated with astrocytoma. Growth factor receptors can also be mutated and abnormally activated, and one example is HER2-neo, which is present in breast cancer. And signal transduction proteins are also important, and they can be mutated and can cause abnormal activation of the growth system, and it can lead to many cancers. Nuclear regulators are also very common, and one very good example is c-MYC, and c-MYC is actually associated with the T translocation of 814. Actually here the translocation occurs from 8 chromosome to 14 chromosome. So 14 chromosome has heavy chain locus for immunoglobulin, and this is an excessively active gene, and if CMYC is transported from 8th chromosome to the 14th chromosome, now it will have excessive expression in the cells who makes the immunoglobulins, and those are lymphocytes, and such as especially B lymphocytes. So it will lead to a B lymphocytic lymphoma, which is called Burkitt lymphoma. Okay, and cell cycle regulators can also be excessively activated, so the regulation can be abnormal, and especially cyclin and cyclin dependent kinase are very important cell cycle regulator, and abnormality in these can lead to melanoma. Okay. So the second group of growth regulator are tumor suppressor gene. So as you can see, their normal job is to suppress formation of tumor. So they actually cause a reduction in growth or an inhibition of growth. So whenever they are mutated, they can be inactivated. So if they are inactivated, this growth inhibition is lost, and the cell can grow without the inhibition excessively. Okay. And among the tumor suppressor genes, the most important is P53 or protein 53 or protein product of 53. It's called the guardian of the genome. The main job of P53 is to detect DNA damage, and after detection, it will actually repair. It will actually help the cell to repair the damaged DNA. So if the damage is severe, P53 can identify it, and P53 will actually call the cell that you are so damaged that you shouldn't live more, you should die now. So it actually gives a suicide signal. So it writes a suicide letter by upregulation of the box, and this box is a suicide letter which actually causes inactivation or disruption of BCL2. So let me explain that. So suppose we have a cell here. Inside cell there are a lot of mitochondria, and suppose this is the mitochondria, and in mitochondria, suppose this is the mitochondria, within mitochondria there is a lot of cytochrome C. Okay, and BCL2 actually is a guard for cytochrome C. BCL2 guards cytochrome C inside the mitochondria. So if BCL2 is inactivated or disrupted, the cytochrome C from the mitochondria will be released into the cytoplasm, and it will actually cause apoptosis. So this is normal. This happens in all our cells if they are damaged by something, some chemical or radiation exposure or something like that. But if P53 is mutated, this sequence cannot occur. So there will be no apoptosis of a very damaged cell, and those cells will actually live up. So though there is severe damage, those cells won't die, and they will form a mass or a tumor. So more than 50% of the tumors or cancers are associated with a mutation of P53, and if germline cells are mutated, it will cause a syndrome called Life-Romany syndrome. It is associated with multiple carcinomas and sarcomas. Okay. So another tumor suppressor gene is RB gene. The main job is it's a carrier, or it's a controller of E2F. So E2F is a important factor in cell cycle regulation. It actually gives a positive signal for cell cycle regulation. So if E2F is bound to RB gene, the cell cycle stops. So RB gene actually holds the E2F in its custody, so it's in its jail, and it stops transition from G phase to S phase. So the cell is arrested in the G phase. So to take E2F out of the custody of RB gene, here comes cyclin and CDK4 complex, and this complex actually bribes the RB gene by giving a phosphate. So phosphate is the bribe for RB gene, and after bribing RB gene with phosphate, it releases E2F, and now E2F can give a positive signal for transition of the cell from a G stage or growth phase to an S phase or synthetic phase. Okay, that's how it works. Okay, the most common cancers associated with RB gene mutations are retinoblastoma and osteosarcoma. So the third group of genes that regulate growth are apoptosis regulator. So I've already mentioned BCL2. It is an anti-apoptotic protein or anti-apoptotic factor which actually seals the mitochondrial membrane to protect the cytochrome C inside it. So if BCL2 ceiling is very highly expressed, so if the BCL2 does not actually release or does not actually cause the cell to die in cases when the cell actually needed to die in case of irreversible damage, so the cells won't die if BCL2 binds too avidly and overexpressed. So the cytochrome C will remain inside mitochondria and the cell will be immortal. Okay, this is the pathogenesis, especially in follicular lymphoma. And what happens is BCL2 is present in 18th chromosome, and here BCL2 comes to 14th chromosome close to immunoglobulin heavy chain locus. And you know, immunoglobulin heavy chain locus is a heavily transcripted or heavily expressed gene, and as BCL2 comes close to immunoglobulin heavy chain gene, it will also be expressed more. So mitochondrial membrane will be more stable and the cell won't die, so the cells will be immortal. So if you express BCL2 more, the cells will be immortal, so cells won't die and they will form a mass, and such as follicular lymphoma. Okay, so as we have gone through some molecular mechanism of tumor development, let's think about some gross tumor progression. So the most common mechanisms of tumor progression in case of malignant tumors are invasion and metastasis. And invasion occurs actually if the cell adhesion or cell-based membrane adhesions or attachments are weak. And metastasis can occur through different routes, through lymphatics, through blood, and through body cavities like peritoneal cavity. So lymphatic metastasis commonly occurs with carcinomas, so carcinomas are commonly associated with it, and hematogenous metastases are associated with sarcomas, okay, CAR-SAR, and seeding in body cavities are commonly associated with GI cancer, most commonly. Okay, next, so clinical features of tumor. If a tumor is a benign tumor, it grows slowly and it actually moves because it's not fixed with underlying structures. In case of malignant tumor, the tumor grows very rapidly and it may be fixed with underlying structure or overlying structures. So cancer cachexia is also a clinical feature of malignancy, characterized by loss of body weight, and the main cause is first of all, a tumor grows very rapidly and it takes up all the nutrition that the person takes, so it actually acts like a parasite. And also sometimes immune response against tumor will produce a lot of TNF, which actually promotes catabolism and leads to loss of body weight. Another type of manifestation of cancer is paraneoplastic syndrome. So this is something special. It's not due to mechanical effect of the primary tumor or not due to mechanical effect of metastasis. Rather, it is due to release of some special or abnormal substances from the tissue that the cancer originated from. Suppose someone has small cell carcinoma, he can have Cushing syndrome due to release of some ACTH-like substance. And if a patient has squamous cell carcinoma, he can develop hypercalcemia due to release of some parathyroid hormone-like substance. Okay, those are not normally secreted from those tissues. Okay, so those are abnormal secretions. So the histologic features of malignancies, neoplasia. If neoplasia is a benign neoplasia, the features will be like that: low nuclear cytoplasmic ratio, such as in a normal cell, you see that the cell is big and nucleus is small. Okay, so there is a ratio where nucleus is small and cell is big. Okay, so nuclear cytoplasmic ratio will be, suppose, 1:4 or 1:6, meaning the cytoplasm will be 4 times bigger or 6 times bigger than nucleus. But in case of malignant tumor, this ratio actually changes. The nucleus actually gets bigger. So in malignant tumor, this nucleus gets bigger and bigger and bigger. So the nuclear cytoplasmic ratio can be as low as 1:1. Okay, so in case of benign tumor, the nuclear cytoplasmic ratio is low. In case of malignant tumor, the nuclear cytoplasmic ratio is close to 1:1. So the ratio is high, and because the cells develops very rapidly, it will have a high mitotic activity. But in case of benign tumor, the cell grows very slowly, so it will be minimally mitotically active. Okay, so mitotic activity will be low here, and mitotic activity will be high in malignant tumor. Okay, Another thing is the cells in benign tumor are actually monomorphic, so they will be same like the normal tissue, and in case of malignant tumor the cells will be of different sizes and shape, so they will have pleomorphism, so pleomorphism of the nucleus or pleomorphism of the cells. And also sometimes you can see some atypical mitosis like Mercedes-Benz logo, like suppose this is the Mercedes-Benz logo. Okay, so you can see something like that, tripolar atypical mitosis, and also there is loss of polarity. Okay, but the one most important thing is benign tumors are well differentiated, very important to know, and malignant tumors are not well differentiated, and that's why they are called anaplasia. So how to find the lineage of the cell? Okay, so this is a big question, especially in a undifferentiated tumor. So if a tumor is undifferentiated, we can't actually histologically say that this tumor came from this tissue, this tumor came from mesenchymal tissue, or this tumor came from epithelial tissue. We can't actually say by seeing it under the microscope. So what we have to do, we have to do some special thing, and they are called immunohistochemical stainings. And for epithelial tissue, we have to stain the tissue with keratin. For mesenchymal tissue, vimentin. For muscle, desmin, so look for the M. For neuroglial tissue, you have to stain them with glial fibrillary acidic protein. And for neurons, you have to stain them with neurofilament. There are some other tissue specific markers also used in histopathology. For prostate specific antigen. For breast, estrogen receptor. For thyroid tissue, thyroglobulin. And for neuroendocrine cells like small cell carcinoma of lungs or carcinoma tumor, you can use chromogranin. And for melanoma, we just commonly use S100 protein. So those are the tissue specific markers, so we use all of those as immunohistochemical staining. Okay, next there are something called tumor markers. So tumor markers are substances that are released from tumor cell into the bloodstream, and we can measure them in the blood. So they are measured from blood, not from histology slide. Okay, so they can be mainly used for actually to detect the recurrence of the tumor, so recurrence, and also to see the response of the treatment. So to look for recurrence and to see response of the treatment. Recurrence and response of the treatment, those two are the main use of tumor markers, but someone say it can also be used for screening of the tumor, but it's not a major use. Okay, major use are those two. And the most common tumor markers are AFP or alpha fetoprotein, which can be present in lung cancer or non-semitomatous testicular tumor, and beta HCG or HCG can be present in choriocarcinoma. Carcinoembryonic agent can be present in any adenocarcinoma of the gastrointestinal tract, and calcitonin can be found in medullary carcinoma of thyroid. Okay, so this is one very important concept is grading. So always remember that grading is done by the lab of a pathologist. Okay, so it's done in the lab. The pathologist don't see the patient, don't know how big the tumor is, don't know anything about the patient. So he just takes a sample of the tissue and just looks up on the microscope. Okay, so that's how he grades it. So grading is actually done to see the maturity or differentiation of the tissue. Okay, if the tissue is well differentiated, we'll call it a low grade, and if the tissue is, or if the tumor is poorly differentiated, we'll call it a high grade tumor. And always remember that if the differentiation is poor, the prognosis will also be poor. Staging. So staging is something done in the hospital. It's not done in the lab, it's done in the hospital, and it's done by an oncologist. He sees the patient, assesses tumor size, what are the nodes that are involved, and look for any metastasis. So it's a clinical evaluation. It's not something pathological. It's a clinical evaluation. Staging is a clinical evaluation, and the most important part of the staging is to see for metastasis, and it's most important for seeing the prognosis. Okay, so this topic is LACL to assess clonality. So I have already mentioned that a tumor actually starts from one cell. So one cell gets abnormally activated and grows excessively, and it forms a tumor. So to assess the clonality, we can do two tests. We can do G6PD isoform testing. So normally there are many isoforms of G6PD and their ratio is 1:1. In case of neoplasia, only one isoform is present. And also in case of B lymphocytes, we can assess the clonality of B lymphocytes by seeing the kappa and lambda ratio. So kappa and lambda are the small fragments of immunoglobulins, and normally kappa lambda ratio is 3:1. In case of a lymphoma where there is a clonal proliferation of the B lymphocyte, this ratio is altered. Maybe it may be reversed, or it may be increased more than 6:1. So that's all about neoplasia today. Thanks for watching the video.